The revolutionary science of CRISPR. CRISPR, or clustered regularly interspaced short palindromic repeats, represents one of the most groundbreaking advancements in genetic engineering. Originally discovered in bacteria as a defense mechanism against viruses, CRISPR is now a tool that allows scientists to precisely edit DNA with unprecedented accuracy and efficiency. By harnessing this bacterial immune system, researchers have unlocked the potential to correct genetic defects, treat diseases, and even reshape entire ecosystems. However, as with any transformative technology, CRISPR also raises profound ethical, scientific, and societal questions. In this video, we will delve into the intricate science behind CRISPR, its applications, and the challenges that lie ahead. What is CRISPR and how does it work? At its core, CRISPR is a natural defense mechanism used by bacteria to fend off viral invaders. When a virus infects a bacterium, the bacterium stores fragments of the viral DNA in its genome, creating a genetic memory. If the same virus attacks again, the bacterium produces RNA molecules that match the viral DNA. These RNA guides pair with a protein called Cas9, forming a powerful complex that can identify and cut the viral DNA, disabling the virus. Scientists have re-engineered this system to target any DNA sequence in any organism. By designing custom RNA guides, they can direct the Cas9 protein to make precise cuts in the genome. This allows for gene editing, deleting, modifying, or inserting genetic material. This revolutionary tool has become the foundation for countless research projects, from treating genetic disorders to modifying crops to improving food security. The discovery of CRISPR, a scientific breakthrough. The path to CRISPR's discovery spans decades of meticulous research and serendipitous findings. In the late 1980s, researchers noticed repeating sequences in bacterial DNA, but had no idea what they were for. Over the years, scientists uncovered that these sequences, along with spacer DNA, played a role in bacterial immunity. The turning point came in 2012, when Jennifer Doudna and Emmanuel Charpentier demonstrated that CRISPR could be harnessed for gene editing in any organism. Their landmark paper outlined how the CRISPR-Cas9 system could be programmed to target specific DNA sequences, effectively creating a tool for precision genetic engineering. This discovery earned them the Nobel Prize in Chemistry in 2020 and ushered in a new era in biotechnology. Applications of CRISPR in medicine. One of the most promising applications of CRISPR lies in the field of medicine. By editing faulty genes, CRISPR holds the potential to cure genetic diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease. In cancer research, CRISPR is being used to engineer immune cells to recognize and attack tumors more effectively. The technology has also enabled the development of gene therapies to combat inherited blindness and certain blood disorders. Beyond treating individual patients, CRISPR has vast implications for global health. For example, researchers are exploring its use to modify the genes of mosquitoes to reduce the spread of diseases like malaria and dengue. However, the medical applications of CRISPR are not without challenges. Delivering CRISPR components to specific cells in the human body remains a major hurdle, and the potential for unintended genetic changes raises concerns about safety and ethics. CRISPR and agriculture, engineering the crops of the future. CRISPR technology is revolutionizing agriculture by enabling scientists to precisely edit the DNA of crops, resulting in improved traits such as higher yields, resistance to pests, and better adaptability to climate change. Unlike traditional genetic modification methods, CRISPR allows for targeted changes without introducing foreign DNA, making it a more efficient and less controversial approach. For example, researchers have developed drought-resistant rice, disease-resistant wheat, and tomatoes with enhanced flavor profiles. CRISPR is also being used to reduce allergens in food crops and increase their nutritional content, such as enhancing the levels of vitamins and minerals in staple crops like cassava and maize. These advancements have the potential to address global food security challenges, ensuring a sustainable and resilient agricultural system for a growing population. Gene drives, controlling ecosystems with CRISPR. One of the most controversial applications of CRISPR is the development of gene drives, a technology designed to spread genetic changes rapidly through populations of organisms. By using CRISPR to introduce genetic modifications that are inherited at a higher than normal rate, scientists aim to control pests, eradicate invasive species, or combat diseases. For example, gene drives are being developed to reduce mosquito populations that transmit malaria or eliminate rodents that threaten island ecosystems. While the potential benefits are immense, gene drives also raise significant ecological and ethical concerns. Releasing modified organisms into the wild could have unpredictable consequences, such as disrupting ecosystems or causing unintended harm to non-target species. Balancing the risks and benefits of this powerful tool remains a topic of intense scientific and public debate. CRISPR and bioethics, navigating moral boundaries. As CRISPR continues to advance, it raises profound ethical questions about the boundaries of genetic engineering. One of the most contentious issues is the potential use of CRISPR to edit human embryos, a process that could eliminate genetic diseases, but also pave the way for so-called designer babies. This raises concerns about equity, access, and the potential for societal division, as only the wealthy might afford such enhancements. Furthermore, the long-term effects of editing the human germline, changes that are passed on to future generations, are still unknown. Ethical guidelines and regulatory frameworks are urgently needed to ensure that CRISPR is used responsibly. The global scientific community is grappling with questions of consent, fairness, and unintended consequences, highlighting the need for careful oversight and inclusive dialogue with society. CRISPR in conservation biology, saving endangered species. CRISPR is emerging as a powerful tool in conservation biology, offering hope for endangered species and biodiversity. By editing the genomes of species threatened by diseases or environmental changes, researchers aim to increase their survival rates. For instance, CRISPR is being used to develop disease-resistant amphibians to combat chytrid fungus, a major threat to global amphibian populations. Similarly, scientists are exploring the possibility of reviving extinct species, such as the woolly mammoth, by editing the DNA of closely related species like the Asian elephant. While these efforts hold great promise, they also raise questions about the role of humans in shaping ecosystems and the ethical implications of de-extinction. The use of CRISPR in conservation highlights both the potential and the challenges of applying genetic technology to address complex environmental problems. CRISPR in medicine, fighting rare genetic disorders. One of CRISPR's most significant breakthroughs lies in its ability to treat rare genetic disorders by correcting the root cause, faulty genes. Disorders like sickle cell anemia, cystic fibrosis, and Duchenne muscular dystrophy, previously considered incurable, are now being targeted with CRISPR-based therapies. By delivering the CRISPR-Cas9 system into cells, scientists can precisely cut out the mutated DNA and replace it with healthy sequences. Early clinical trials for conditions like sickle cell anemia and beta thalassemia have shown remarkable success, with patients experiencing significant symptom relief or complete remission. While these advancements mark a major step forward, challenges such as off-target effects, efficient delivery mechanisms, and equitable access to these treatments remain critical areas of focus. CRISPR and cancer therapy, a new frontier in oncology. CRISPR is transforming cancer treatment by enabling researchers to reprogram immune cells to better target and destroy tumors. A prominent application is the engineering of T cells, a type of immune cell, to recognize and attack specific cancer cells. This approach, known as CAR T therapy, has shown promise in treating blood cancers such as leukemia and lymphoma. Additionally, CRISPR is being used to identify genetic vulnerabilities in tumors, helping scientists develop more effective drugs. Another groundbreaking application is the potential to prevent cancer by editing genes associated with high cancer risk, such as BRCA1 and BRCA2. While these innovations offer hope, they also come with challenges, including ensuring the safety and precision of the edits and addressing ethical concerns about preventative genetic modifications. CRISPR and infectious diseases battling pathogens. CRISPR has opened new possibilities in combating infectious diseases by targeting and disabling the DNA or RNA of harmful pathogens. For example, CRISPR has been used to develop antiviral therapies against diseases like HIV, hepatitis B, and herpes by directly editing the viral genomes to render them inactive. Researchers are also leveraging CRISPR to create diagnostic tools, such as CRISPR-based tests for COVID-19, which provide rapid and accurate results. Furthermore, CRISPR is being explored as a tool to combat antibiotic resistance by designing systems that can selectively kill drug-resistant bacteria. These advancements not only offer new treatment options, but also provide tools to monitor and prevent future outbreaks. However, the potential for unintended consequences, such as off-target effects on beneficial microbes, highlights the need for careful research and regulation. CRISPR and human longevity, editing the aging process. CRISPR is beginning to unlock the secrets of aging by enabling scientists to study and potentially modify the genetic factors that drive the aging process. Researchers are investigating how CRISPR can be used to repair or reverse damage to DNA that accumulates over time, a major contributor to aging and age-related diseases. For instance, studies in mice have demonstrated the ability to extend lifespan by editing genes associated with cellular senescence and oxidative stress. CRISPR is also being used to enhance the regenerative capacity of stem cells, paving the way for therapies that could slow aging or even rejuvenate tissues. While the concept of editing the human lifespan is still in its infancy, it raises profound ethical and societal questions about the implications of significantly extending human life and the potential inequalities such advancements might create. CRISPR and agricultural revolution, engineering climate-resilient crops. CRISPR technology is revolutionizing agriculture by enabling scientists to create crops that are more resilient to climate change, pests, and diseases. By editing genes responsible for drought tolerance, heat resistance, or nutrient efficiency, researchers are developing plants that can thrive in harsh environments and ensure food security for a growing global population. For example, CRISPR has been used to engineer rice varieties with higher yields and wheat resistant to fungal infections. It has also enabled the reduction of allergens in peanuts and the removal of harmful compounds in crops like cassava. While these advancements promise to transform farming, concerns about biodiversity, the impact on ecosystems, and public acceptance of genetically modified crops remain critical issues to address. CRISPR and synthetic biology. Designing new life forms, CRISPR is unlocking the potential of synthetic biology by allowing scientists to design and create entirely new organisms with tailored genetic traits. This includes microbes engineered to produce biofuels, biodegradable plastics, and even pharmaceuticals. Synthetic biology powered by CRISPR also opens doors to creating plants that absorb more carbon dioxide or generate their own fertilizers, offering innovative solutions to environmental challenges. Beyond practical applications, CRISPR is being used to explore the origins of life by assembling synthetic cells from scratch. While this field holds incredible promise, it also raises ethical and safety concerns, such as the potential misuse of engineered organisms or unintended consequences in natural ecosystems. CRISPR and bioethics, navigating the moral dilemmas of genetic editing. The rapid advancement of CRISPR technology has sparked intense ethical debates about the implications of editing human DNA. Key concerns include the potential misuse of CRISPR for non-therapeutic purposes, such as creating designer babies with enhanced physical or cognitive traits, and the possibility of exacerbating social inequalities. The controversy surrounding the first genetically edited babies in 2018 highlighted the need for global consensus on ethical guidelines and strict regulatory frameworks. Additionally, questions about consent, access, and the long-term impact of genetic edits on future generations remain unresolved. As CRISPR continues to evolve, it is essential for scientists, policymakers, and society to work together to ensure responsible and equitable use of this powerful technology. The future of CRISPR, expanding beyond genetics. CRISPR's potential extends far beyond its current applications, with scientists exploring innovative uses that could redefine medicine, biology, and technology. Emerging developments include CRISPR systems that can edit RNA instead of DNA, offering new treatments for diseases without permanent genetic changes. Researchers are also working on CRISPR-based tools for precise epigenetic editing, which could regulate gene expression without altering the DNA sequence. In addition, CRISPR is being integrated with advanced technologies like AI and nanotechnology to enhance its precision and efficiency. As we look to the future, the possibilities for CRISPR seem limitless, but realizing its full potential will require overcoming technical challenges, addressing ethical concerns, and ensuring that its benefits are accessible to all. CRISPR technology is not just a scientific breakthrough, it's a game changer for humanity. As we stand at the threshold of this genetic revolution, the possibilities are endless, from curing diseases and fighting climate change to reshaping agriculture and creating synthetic life forms. But with great power comes great responsibility. The ethical dilemmas, safety concerns, and equitable access to these innovations demand our collective attention and action. Join the conversation and be part of shaping the future of CRISPR technology. Share your thoughts, ideas, and questions in the comments below. Don't forget to like this video if you found it informative and subscribe for more in-depth explorations of groundbreaking technologies. Together, we can ensure that CRISPR's immense potential is harnessed for the betterment of all humanity. The future is being written in our DNA. Let's make it a story worth telling.